I don't know if we're going to see Michael or not, but we'll find out. All right, y'all. So let's do this thing. This is our last lesson of Unit 3. Woo-hoo. And you see you've got your packets. Everybody has everything they need now for Units 4 and 5 and right up till Christmas break. So James, you can sit in the chair, sweetheart, really. You really just like to stand? Okay. Yeah, there's a nice cushy chair right there. You can just kick the bag out, whoever bag. All right, so we're talking about cell division today, which is why this was a good lab, because obviously if these dudes are going to regenerate, there's going to be a lot of cell division going on. So let's see what we've got. So we're going to be discussing predominantly mitosis, which is the process of cell division for the purpose of growth and development. And in a week or so, we're going to get into meiosis, which is cell division for the purpose of producing gametes or sex cells. So cell growth, division, and reproduction is where we're headed today. Ozzy, you come here because people have food. Come on, good boy. There you go. So there are limits to cell size. All right, why are there limits to cell size? Well, you've got to deal with a surface to volume ratio when you're talking about any kind of like functioning machine system. Okay, so when you get a cell that gets bigger and bigger, you can think about it blowing up a balloon, right? So you've got, say you've got a balloon and it's this big. If you were to maybe take the air and figure out how many cubic centimeters of air you had and figure out how many cubic centimeters of latex you had, there would be a point where the ratio becomes too much for that balloon to handle, right? Where you don't have enough latex to contain the air because the pressure has gotten too high. In this case, what it is the processes are too involved. You've got cells that have all these internal processes going on, right? They're replicating proteins and synthesizing proteins, replicating RNA and DNA, and they're doing all of the functions that a cell does, and they're growing at the same time. When they get to a point where it's too difficult to exchange materials, waste, and things they need across their membrane, they have to divide or they just can't keep up. So we have our cell cycle, all right, and this is like an overview of the cell cycle, but we're going to break it down into phases. So this is how cells grow and divide as they follow this cycle. So you've got sexual reproduction and asexual reproduction when you're talking about division. We're going to touch on this lightly, and then next week when we really get into meiosis and gamete production and stuff, we talk about it a bit more. But with sexual reproduction, you're going to produce genetically, wait, that's in the wrong spot. That's supposed to be on the other side. It's supposed to be on the other side. Oh my gosh, look at that. Okay, hold on, wait a minute. That is very wrong. Look at that. But I caught it, not you.
No, in asexual, both your parents.
Yeah, because I caught it. I just caught it. Look at that. I just had my headings in the wrong place. Now they're
No,
fixed.
there's another brownie point.
Where?
On the right.
It's actually
Both are. You got it. There you go. Good call. Sorry, man. He does want brownies. All right. So asexual reproduction. All right. In asexual reproduction, you're producing genetically identical organisms. So this is what happens in prokaryotic cells. Do you remember what is the difference between a prokaryotic and a eukaryotic cell? Yeah, James. Yeah. Your eukaryotic cell has nucleus with a nucleus inside of a nuclear membrane, right? And that's where your DNA is kept. Prokaryotic cells are more ancient, and they just sort of have their DNA floating around in their cytoplasm, right? So asexual reproduction is going to produce genetically identical organisms, and that is what happens in prokaryotic cells. It also happens in some eukaryotic cells. So predominantly, it's going to occur in single-celled organisms. So tiny single-celled organisms, sometimes things like bacteria, right? Also things like yeast. Last week when we had the yeast. So it allows for rapid reproduction in organisms in favorable environments. So here's what we'll talk a lot about when we move into the concept of natural selection and asexual reproduction versus sexual reproduction. So are y'all good over there? Okay, so if you reproduce asexually and you are in an environment that is very favorable to you and all your traits, it is very advantageous to you to just keep on reproducing. You don't have to find a mate, right? You're going to pass on your DNA real easily, usually by just splitting yourself in two. And you're just going to keep on thriving, and your DNA is going to get passed down to further generations because your environment is very favorable. If something in your environment changes, however, and now you are no longer well suited for that environment, your entire genetic line can just disappear, which is why sexual reproduction is advantageous, because it allows for differences, sometimes just small differences. And those differences can be enough to keep an organism's genetic material in the gene pool to keep getting passed to other generations. So with sexual reproduction, you've got organisms with genetic information from two parents. And this sometimes happens, it's not just animals, there are plants that can reproduce both sexually and asexually. And then most animals are going to reproduce sexually, but not all animals. There are going to be some that don't. So it occurs in most multi-celled organisms, including plants and animals, and it increases genetic diversity for changing environments, which is what we were just saying. So think about how, like, a great example is bacteria that divide asexually, and you take an antibiotic, and what happens to that bacteria? It's gone, right? It's gone. As long as it's all genetically identical, you can get rid of it. So that's a great example of where asexual reproduction is not advantageous to the bacteria, but it's advantageous to us if we're the ones that are infected. So cell division. What are y'all doing over there?
They're passing my backpack around a line.
Oh, this is yours.
All right.
His.
So here's our prokaryotic cell division. All right, so if you look here, we have some little single-celled organisms, right? And this is how prokaryotic cells divide. This is also how a lot of very tiny eukaryotic cells, they look similar when they divide, but the process is actually different. So when your prokaryotic cells divide, it's called binary fission. So what's fission mean? Who knows fission?
Together? Oh, no, apart.
Apart. What would be the
Fusion's together,
opposite?
fission.
Yeah, fusion would be putting it together, and fission would be breaking it apart. And binary would mean into two. So binary is like in computers, they use binary language, right? It's 1, 0 on off, and there are multiple ways to go about using that code basically to create sort of like the way DNA works. You code for things. Actually, if my husband comes back down, I'll show him I'll ask him to count for you. He was demonstrating the other night how to count in binary. You can get to 6, I think. And it was Drew was cracking up.
I think I can also do it.
Can you do it? Yeah. Do you remember how to do it?
1, 1, 0, 1, 1.
No, I mean like on your fingers. Can you do it on your fingers? Oh,
Well,
so it's very entertaining. I'll have him do it when he comes back because I'm sure he'll be back. It's a lot of fun.
1, 1.
Good job.
All right. So yes. So in your prokaryotic cells, your chromosome is a single DNA molecule that first replicates, attaches a copy to different parts of the cell membrane, and then the membrane pulls apart and the chromosomes are separated. Jay, so you want to call or are you just sitting there? Hey, can you demonstrate how to count in binary? We were just talking about binary fission, and Drew tried to do it in his class, but he couldn't remember it. He's getting cheese and sort of half listening to me.
Why do you want to count binary?
Because it's a good example. Can you come over here though? Because Michael's on there.
Oh, we get this paper. It's easy.
No, do your fingers. Like at dinner.
No.
Alex doesn't know. Alex can count it, but he can't do it on the fingers. All right, he's gonna demonstrate.
On camera?
Yeah, you're on camera because Michael's on
I know
camera.
what you want me to do.
Yeah, just count it.
All right, so this is 0. So if you're binary, you only have 2 fingers, right? That's it. So we have 5 on each hand because we're humans and we count to 10. That's why everything is base 10. We have 10 fingers. we lived in a world where we had 2 fingers, that's all we had to count on, right? So 0, 1, 2,
That's I do.
3,
That's why Drew wanted to remember it.
and then it would be 4. And then, 5, and then 6, and then
I think 6
7.
is probably 7. Okay, excellent.
2, 0, 2, 1, 2, 2, 2, 3, 8. 8's the highest you can go.
8, there you go.
So that's 8.
Oh, thank you.
That makes so much more sense. I was
Yay.
like, why would Drew be cracking up?
Yes, Drew wanted to count it on his
Yeah.
fingers, but he just couldn't make it work. Thank you, Jason. There you go. Great. This is why, yeah. Telecommunications engineers are fun. All right, so there we go. So yes, your prokaryotic cells are just going to have their DNA replicate. It's going to stick to sides of the membrane. The membrane is going to pull apart and just squish apart in the middle. Is he in the waiting room again? There he is. Hooray. He's made it. Awesome.
He didn't get to see that.
Well, he can watch it on the record. Yeah. So you have to tell him to go back and watch that.
That was
Yeah.
hilarious.
So Michael, you're going to have to rewind and watch what you just missed because it was entertaining. So your step 1 is going to be your DNA replication. And then after that, you're going to have your chromosome segregation where the two chromosomes segregate or separate and move to opposite ends. And then call those poles, just like it were a globe. And then the whole cell separates. Eukaryotic cell division is way more complex. So with eukaryotic cell division, it occurs in stages, and the overarching stages are going to be interphase and mitotic phase. Now, interphase is, you'd like to think of it as the cell at rest, but it's really not at rest because it's doing all of its regular processes. So it's very busy, but it's not dividing. It's not in the process of dividing. Mitotic phase is all of the processes of dividing, and that has multiple steps. Now, I asked the other group to come up with a mnemonic device for remembering the steps, like, you know, King Henry died unusually drinking chocolate milk, or please excuse my dear Aunt Sally. So I'll have to share with you some of the ones they came up with because they were really interesting. They were yeah, they're entertaining. So interphase, like I said, not really at rest, but it's when the cell is just hanging in between. It's doing all of its normal stuff, but it's not dividing. So in interphase, the activities are indispensable, right? This is the stuff that it has to do before it gets to the next mitotic phase. So interphase generally lasts 12 to 24 hours in mammalian tissue. So you have a cell in your tissue, depends on which kind of tissue, but you have a cell in your tissue that divides, and it divides again probably a day later. They don't stay long. It depends on the tissue. So you've got some cells in your body that almost never divide, things like neurons in adults, okay? But then you have some cells that divide super rapidly, like skin cells, because they're constantly being sloughed off. And so like plants can go through the whole process of dividing itself. The actual mitotic process in plants can be like 10 to 20 hours. In humans, like I said, there's some cells that almost never divide. There are some that are going to take a day, and there are ones like on your skin that are the fastest ones, and sometimes it's like an hour and a half, and you've already got a whole cell division process done. So it moves very quickly. So by studying molecular events in cells, scientists determined that interphase can be divided into 4 steps. So this is interphase steps. These are the ones that are less important for you to like totally memorize. I want you to know that things go on in interphase, but it's the mitotic phase that's going to be most important for you to know. So they call them gap 0, gap 1, synthesis, and gap 2. So in gap 0, so here is an example of a neuron. This is a kind of cell that's not going to divide very often at all. So there are times when a cell will leave the cycle and quit dividing. This may be a temporary resting period or more permanent. So in the case of a neuron, it's going to be more permanent. An example, okay, I know you see whatever this is right here. It's actually covered up, and I didn't know it was. Yeah, the picture's in my way. Don't know why. There.
There we go.
Does that sound
Better. No. That's not a brownie point. So, an example of the latter is a cell that's reached an end stage of development and will no longer divide like a neuron. Why don't you want your neurons to divide?
You don't want them?
Yeah, why would you want your neurons to stay put?
Okay, so connecting together?
Yeah.
Only wrap your nervous system.
Yeah, these are what help build pathways to memories, knowledge, information, everything your body does as far as movement.
Information lines, so want to stay the way they are.
Right. If they get messed up, it can cause some real concerns, right? So the G1 phase, G1 phase is when you've got the first 4 phases of the cell cycle take place in eukaryotic cell division. This is the part of interphase where cell moves through a series of steps in an orderly manner. So cells increase in size in G1. They produce RNA and they start to synthesize protein. An important cycle control mechanism is activated here. All right, so you're going to find that throughout the cell cycle, there are what they call checkpoints. And it's literally like, it's exactly what it sounds like. So your cell's running through all these processes and there's a point where it's programmed to stop, check, make sure there's no mistakes and keep going, which is really a complex thing to have evolved in a system like that. That's very complex. So after your G phase, G1 phase, you're going to go to S, and this is your synthesis phase. So this is when you're going to produce 2 daughter cells. So you must have complete DNA instructions in your cell, and it must be all duplicated and ready to go. So this is where your DNA replication starts, okay? And then you've got your G2 phase. And this involves further growth and protein synthesis during the gap between DNA synthesis and mitosis. So the cell will continue to grow, produce new proteins. At the end of the gap, another control checkpoint is going to come in and determine if the cell can divide or if it needs to wait. And this is kind of one of your last checkpoints before you start the actual process of dividing. So here's where we get into the actual mitotic phase. Here's where all the dividing begins. What do y'all laugh at? Red jelly beans. You know what they look like? Mike and Ike, but they're not.
Jack just said, look, there's Cheetos.
Oh my gosh, they do look like Cheetos. You're right.
The last picture.
Oh yeah, they did. So I spent time to make some computer animations for these cell divisions because I drew them once and drew something very embarrassing. So
What you drink?
it has to do with the last step, and when I get to it, you might have an idea of what I drew. But it was, yeah, I still have students that I taught 15, 16 years ago that remember it. So if that tells you anything.
Oh, yeah.
All right.
You told so many stories.
Hey, you were the one that, yeah. All right, so cell growth and protein production stop at this stage. We're not growing anymore. We're going to divide, okay? And all the cell's energy starts to focus on this whole complex process of going from one parent cell to what we call 2 daughter cells. So mitosis is much shorter than interphase, lasting perhaps only 1 to 2 hours. So you've got cells that are dividing, like we said here, sometimes only 1 to 2 hours, sometimes longer. Sometimes it takes a longer time for the cells to actually divide. And sometimes they sit in interphase for a much longer period of time. So it just depends on where the cells are, whether we're talking about human body cells, plant cells, you know, there's obviously, all kinds of cells. So they're all going to have different needs where that's concerned. So mitosis gets divided into the following phases. And these are the ones you need to absolutely memorize the order of and know what happens in them. So you got prophase, metaphase, anaphase, and telophase with cytokinesis. Telophase sort of implies that cytokinesis comes, but your book made a point of pointing it out, and so did the other guy's book. So if you have interphase first, a lot of people have memorized PMAT. That's what they've gone with, is PMAT. And they just remember that there's an I at the front and then a C. You don't really need the C because, like I said, cytokinesis is kind of the end part of telophase. But I had the other guys come up with some mnemonic devices. So they came up with, let me see, Colson came up with, I play Minecraft at the computer. That was a good one. Drew's was, I punched my annoying tennis coach. That was memorable, right? It's memorable. If you remember it, then that's the point. Let's see, wait a minute. There were 2 more. Lewis's was, shoot. I can't remember what Lewis's was. It was good. His was the most sensible one. Let me put it that way. Darcy's
Not hard.
was improper poopers must always think clean, which I loved. And Drew commented back on it with what's an improper pooper. So they were in their, in their discussion in their portal, and it was entertaining. What was Lewis's? Shoot. Important people must always something. Important people must always something. So you can come up with whatever works for you, but you're going to want to remember their order, and you're going to want to remember what happens in them. So here's your
If you go down Hannah Lane, you find out what.
So here's your mitosis, your first phase, your prophase, your P, your pooper or punched or whatever you're going to remember. All right. So during prophase, the complex of DNA and proteins contained in the nucleus, known as chromatin, is going to condense. So chromatin coils and becomes increasingly compact, which results in the formation of visible chromosomes. Sister chromatids are pairs of identical copies of DNA joined at a point called a centromere. All right, this whole thing here is a chromosome. This branch is a chromatid. This branch is a chromatid. And in the middle is the centromere. Okay, and that's where they're joined. It's like some protein bonds that stick them together. So the main things that happen in this phase, and this is the part you're going to want to come back and look at these slides where it says your main events are, this is stuff in purple, what you're going to remember. Condensation of chromosomes, movement of centromeres, formation of mitotic spindle, and the breakdown of nucleoli. So obviously, if you're going to start moving your chromosomes around, you're going to have to break down your nuclear membrane because they have to come out in order to divide. So that nuclear membrane that separates these eukaryotic cells from prokaryotic cells is going to break down and disappear. Now, this mitotic spindle it references, that's these things right here. You see these? These are fibers. Do you remember when we said cells contain a cytoskeleton and you don't really see it? It's just like there's kind of a structure within it, but you can't really see that cytoskeleton. Well, this is when you see the cytoskeleton. It actually moves and shifts. So I think I told you guys before, the cytoskeleton is kind of like a knit blanket where all those fibers are there, but they're flexible, but it still maintains some shape. Kind of like if you were to throw a blanket out, it kind of maintains some shape, but you can still mush it. Well, this would be like if it started pulling apart, all right? So then the next phase here is your metaphase. And in metaphase, this is when these chromosomes actually line up. And it's really pretty cool because these spindles here are gonna force them all to the center, okay? Shove them into the center like that, and they're going to line up down what they call the metaphase plate, or sometimes I'll call it what's the other word for it? It's not coming to me at the moment, but it's a metastatic plate. So this is your third phase, and this is where your duplicated genetic material is lined up nicely right here. So you have these long protein filaments called kinetochore microtubules, and they extend from the poles at either end. So look, now we have poles, all right? Exactly like a globe. Yeah.
Is the final sentence supposed to be in past tense?
Yeah, it doesn't matter. I didn't write it. I copied it. So copying stuff. So main events in metaphase. Your mitotic spindle is now fully developed because it's pushed them to the center. Your centromeres are at opposite poles of the cell, and your chromosomes are lined up at your metaphase plate. So think about these guys right here. These centromeres here, or centrosomes, sometimes are called centrosomes, sometimes are called centromeres. They are not visible at all until your cell starts to divide. They're always there, but they're not visible. They're barrel shaped, and they're where all these little spindles connect, okay? And they suddenly become visible when your cell division process starts, which is kind of cool. Yeah.
I think what he pointed out, it says and attach. I
Extended.
think it's supposed to say and are attached.
Could be. Yeah, that could be one. I'll give you two. I'll give you two.
Give James the credit.
Yeah. All right. So anaphase. This is where we are next. So here, I'll play that one more time for you,
No, that's.
because some of y'all weren't looking. So here's anaphase. Watch. So what happens here in anaphase is this is your fourth phase, and this is when they start to separate. So now you've got these poles, right? And they're just like, think of them almost like magnetic poles.
Oh,
And you've got your chromosomes lined up like X's all down the middle. And then these fibers, which are attached to them, first they pushed them to the middle, and now they're going to pull them to the poles. And as they pull them to the poles like that, that little centrosome that was holding the two sister chromatids together is going to get broken. That bond gets broken. And you can tell they're being pulled. See how they're shaped? No, I'm leaving you, right? They're getting pulled away. So,
so I say 4 kidney.
and don't worry about the dot, dot, dot. It just means there was more in there that I took out.
2 kidney.
So cohesion proteins break down that we're binding the sister chromatids together. Your sister chromatids are now, your little chromosomes are pulled towards opposite poles, and your non-kinetic core spindle fibers lengthen, elongating the cell. So your cell now starts to stretch too. So then you have your telophase. All right, now telophase is when you're, now it's not quite broken, it's peanut shaped, right? It's sort of peanut shaped, but all of your chromosomes and your genetic material are still kind of just floating around in cytoplasm. So your nuclear membrane has to sort of reform and encapsulate all of that data, get it back where it goes. Now, meanwhile, all of your organelles are getting divided up too between these cells as they pull apart. So you have to remember all those are there too, right? Your ribosomes are there, your endoplasmic reticulum, any vacuoles, all that stuff is in there, and it's all getting divided up. And there's a whole thing you could do. You could study that for a year. I mean, really, you could. So along with telophase, the cell undergoes a process called cytokinesis, which we're going to look at next. And that's when the cytoplasm actually splits apart. So your chromosomes begin to decondense, your spindle breaks down, and your nuclear membrane reform. All right, so here's your nuclear membrane reforming. These guys are in here, and they're eventually going to unravel again like they were before. So you have to think about it. Your DNA is in your nucleus, and it's just sort of like yarn, just kind of all tossed in. And at the beginning, it got itself all wound up and organized, separated, and now it's kind of again back where it was. So this is cytokinesis, and this is where I drew the embarrassing thing years ago that I tried to avoid. And you can probably guess what I drew. So this line is called cleavage. And I drew something that looked a lot like this, drew a line down the middle and said, and cleavage. And my whole class started laughing, and I realized what it looked like. And it looked exactly like you, if you haven't at all a dirty gutter mind, you know what I've drawn. And everyone laughed, and I was mortified. So in cytokinesis, that's when your cells physically separate. And the way this happens is you have a contractile ring, okay? And it comes in between the two cells, and it just shrinks right down and pinches them off. Just like if you were to squeeze a hose or a tube, just shrinks it down. Y'all over there. So it's called a cleavage furrow, and then it separates. Now, it's actually composed of actin, which this video is going to show you in just a second here, that contractile ring is composed of actin, which is one of the 2 major proteins that help your muscles function. So go figure, it's also contracts. Hey, Mom. All right, so we're gonna watch this one on cell cycle. This is good animation here.
There he is.
An adult human contains an estimated 100 trillion cells.
Y'all over there,
and
pay
yet we
attention.
start life
Jack,
as a
Jim.
single cell. To grow, develop, and repair tissue damage, we rely on cell division. In eukaryotic cells, this process is accomplished by a series of well-orchestrated steps called mitosis. Every day, our bodies must produce millions of skin cells to replace those lost through normal activity. Each of these cells must have a complete complement of the genetic material. Prior to cell division, where the cell divides into 2 identical cells, the DNA needs to be replicated so that each daughter cell receives an exact copy. Following DNA replication, the chromosomes condense in the nucleus of the cell. DNA condenses by wrapping around cores of histone proteins, forming nucleosomes. This beads on a string structure is called chromatin. As a cell prepares to divide, chromatin coils up further, shortening and condensing the chromosome. The replicated chromosomes are called sister chromatids. The replication of DNA and the formation of sister chromatids is one part of the entire cell cycle. To prepare for cell division, the cell goes through interphase, which can be divided into 3 distinct phases. During G1 or GAP1 phase, all the organelles and cytoplasmic components, including the centrioles in animal cells, replicate. Then during S or synthesis phase, the DNA replicates. Finally, during G2 or GAP2 phase, all the enzymes needed to aid in the process of cell division are produced. Most eukaryotic cells spend a great deal of time in interphase and a very short period of time actually dividing, a process called mitosis. The cell is now ready to go through mitosis, which consists of prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes condense and become visible, appearing as 2 sister chromatids held together at the centromere. The cytoskeleton disassembles as the spindle begins to form. In animal cells, centrioles play an important role in the distribution of the chromosomes in the dividing cell. The centrioles migrate to opposite poles, establishing a bridge of microtubules called the spindle apparatus, and the nuclear envelope breaks down. Towards the end of prophase, chromosomes attach by proteins in their centromeres called kinetochores to microtubules from each pole, moving the chromosomes toward the equator of the cell. During metaphase, all chromosomes are aligned at the equator of the cell, called the metaphase plate. Anaphase begins with the degradation of proteins that hold sister chromatids together, freeing individual chromosomes. The free chromosomes are then pulled by their kinetochores to opposite poles. At telophase, a cleavage furrow forms in the center of the cell. This indentation is made from a constricting belt of actin filaments surrounding the inside of the cell's circumference. Chromosomes cluster at opposite poles and begin decondensing as the nuclear envelope reforms around them. The spindle apparatus disassembles as the microtubules are broken down into tubulin monomers that can be used to form the cytoskeleton of the daughter cells. In animal cells, cytokinesis completes cell division by extending the cleavage furrow to completely separate the newly formed daughter cells. Since plant cell walls cannot be constricted by actin fibers, vesicles form an expanding membrane partition called the cell plate. Like animal cells, plant cells use cytokinesis to finish the division of the contents of the cytoplasm between the 2 identical daughter cells. During the cell cycle, certain checkpoints are encountered to make sure the process is occurring accurately. And if it is not, the cell cycle will stop at the checkpoint and correct, or possibly inhibit that cell from dividing. The first checkpoint is the G1S checkpoint and is considered the primary point at which the cell cycle continues or stops. External signals and growth factors can influence the cell cycle and affect the progress at or before this critical checkpoint. The G2M checkpoint allows cells that have successfully completed all 3 phases of interphase to begin mitosis. The last checkpoint is the spindle checkpoint, ensuring that all chromosomes have attached to the spindle in preparation for anaphase. Growth factors, the size of the cell, and the nutritional state of the cell are all contributing factors in cell cycle regulation, ensuring that only certain cells divide at appropriate times. Once all the checkpoints in interphase are cleared, mitosis can occur. From interphase to cytokinesis, the entire process of cell division can take on average 10 to 20 hours in a typical plant or animal cell. Depending on the nature and use of the cell, the process can happen at different frequencies as well. In humans, our skin cells have a high turnover rate due to wear and tear and go through mitosis very frequently, whereas other cells, such as adult neurons and muscle cells, rarely divide. The accuracy of mitosis, as well as the consistency
why they
of the
slap those up
checkpoints
so
during
fast.
interphase, ensure that most cells in a eukaryotic organism can produce identical copies of themselves.
It's like,
This
this-
process allows for growth and repair to prolong overall physiology as well as life itself.
Come there we go. That's a good one.
Professor Dave.
Good
An
graphics.
adult human
Professor Dave has
contains
one,
an-
but it's long.
Eh,
Very long. So what are ways that we regulate the cycle? We were just talking about how there are various checkpoints along the way, right? So we have controls on cell division. If we didn't have controls on cell division, we'd have a real big problem, which we're going to discuss in a minute. So there are dozens of proteins and external factors involved in regulating the cell cycle. Cyclins are among the most important core cell cycle regulators. Cyclins are a group of related proteins. As the name suggests, each cyclin is associated with a particular part of the cycle. There you go. And so these are the proteins that actually control those checkpoints that we talked about a minute ago.
Spicy Cheetos.
Spicy Cheetos, there you go. Careful, Cole. The table bonked.
Mountain Dew
Again.
sting.
So cyclins drive the events of the cell by partnering with a family of enzymes called cyclin-dependent kinases, or CDKs. And then a lone a lone CDK is inactive, but when it binds with another one, it becomes functional. So this is the actual chemical processes that go on to keep the cell cycle regulated. So a famous example of how cyclins and CDKs work together to control cell cycle transitions is that of maturation promoting factor, MPF. The name dates back to the 1970s when researchers found that cells in the M phase, in mitosis phase, right, contained an unknown factor that could force frog egg cells stuck in G22 to start. So this is this whole, like it starts this part of the phase and another phase, subscript 2, N subscript, all these little ones, and it's hard for me to write out. So to enter the M phase. This mystery molecule called MPF was discovered in the 1980s to be a CDK bound to its M cyclin partner. So for years, scientists have been trying to determine why cells divide when they divide, what stops them, and why they die. They die in a process called apoptosis. It pops. It pop. No, they don't always pop. Anyway, it's genetically controlled response, and this is how they undergo programmed cell death. So in your DNA is information to tell your cells to divide, to create cyclin proteins that will bind with specific other cyclin proteins at specific points in the cell division process to make sure that you don't have any mistakes along the way, and also to die at a certain time, that's it, we're gonna die. And so it's a phenomenon that plays a major role in development pathways and provides homeostatic balance of cell populations, and it's regulated in many diseases, including cancer. So this is why apoptosis is so important and why cyclins are so important. So we're going to watch this on cancer real quick. This is just a graphic. This shows, this is how cancer happens.
I don't like that sad.
Right? They made it creepy. But what you got here are cells that are not dying. They're just continuing to divide. And look at the cells around them. They're not dividing at the same rate.
Like somebody gave a really creative-
It's a great, I don't know who the Foley artist is for this, but great job.
like,
Yeah.
So these are cells growing without those checkpoints to stop, without programmed cell death. And this is what causes cancer. That would be how a tumor grows. That's exactly how a tumor grows. Now we have a more specific video we're going to watch in a second that gets more in depth into this process, but this was just such cool graphics. It was worth watching.
still not sad.
Yeah, the sounds are creepy. But here's the thing, that first cell that divides inappropriately then is going to pass on that information to all of its offspring. But that first, the minute a cancer cell is born, basically the one that is not programmed to die or that is programmed to divide improperly, that can happen years and years and years before anybody ever finds cancer itself, like the disease, and finds that you have cancer or somebody has cancer. So this explains it pretty
Normal
in depth.
cell to a cancer cell. Take 5 minutes to get the picture. More than 60,000 billion cells make up the human body. They are the units that form the tissues which make up our organs, like the liver, heart, and lungs. When the body needs it, our cells split into two and replace those cells that are defective or have come to the end of their lifespan. This makes it possible for our tissues to preserve their shape and their respective functions with the passage of time. Every cell is therefore programmed to multiply and die. This ordered but complex program is controlled by the center of the cell, the nucleus, which contains chromosomes containing many genes made up of DNA. Sometimes some of these genes undergo a change. The nucleus then sends out abnormal orders and the cell goes wrong. It multiplies uncontrollably and takes on a life of its own. Each new cell produced contains the same defect. The cells proliferate chaotically and form a tumor. This process may be short, but is often long. 10 to 30 years may separate the birth of a first abnormal cell from the appearance of a tumor of about 1 cubic centimeter. The tumor forms many blood vessels in order to survive. These will supply the tumor with oxygen and nutriments, allowing it to live and grow.
Very British.
This is what we call the phenomenon of angiogenesis. The tumor, however, only really becomes dangerous when the cancerous cells begin to invade through the vessels into the adjacent areas and spread to the surrounding organs.
Well, that's not good.
These cells can then invade other parts of the body, multiply and produce new tumors. Metastasis is the term used for this spreading process. But why does a cell become cancerous? There are many factors, and they can exist to different degrees. Factors have been identified, such as hereditary genetic anomalies, exposure to some viruses, HIV, hepatitis B, C, and D, papillomavirus, Epstein-Barr virus, exposure to toxic agents, chemical products, radiation, sun, unhealthy behaviors, such as the consumption of alcohol and tobacco, or a diet too rich in fat and low in fruit and vegetables. Today, over 25 million people in the world live with cancer. 7 million die from it each year. The disease represents the leading cause of mortality for those under 65. Lung cancer is the most widespread, followed by breast cancer, colorectal, stomach, and prostate cancer. The means to combat cancer do, however, exist. These often complementary treatments are used alone or in combination according to the type of cancer and its status. The goal of the treatments is to make it possible to eliminate the tumor and cure the patient in early stage cancer, or like a chronic disease, to control its development. There are mainly 3 kinds of treatment. Surgery consists of removing the tumor in part or in its entirety. Radiotherapy exposes the tumor to radioactive rays, which prevent the diseased cells from multiplying and destroys them. Chemotherapy consists of administering systemic medicinal substances to destroy the cancerous cells or prevent them from spreading. Chemotherapy and radiotherapy act on cancerous cells, but also on healthy cells, which explains their side effects. But today, better knowledge of the characteristics of cancerous cells makes it possible to develop therapies that target the cancer development mechanisms more specifically. Sanofi Aventis R&D is already present in all these areas to fight against cancer on all fronts.
So if you have cancer in a particular organ, okay, like for example, my uncle had kidney cancer and it became metastatic, which means it spread, right? That means that every cancer cell in his body, everything in his body that was at this point cancerous, when it got into his bones and it got into his brain, those cells are actually kidney cells that have been misplaced. So if you were to go in and take those cells out and look at them under a microscope and do whatever test you needed to do to find out what kind of tissue it is, sometimes it's just looking at it. You could tell where they came from potentially. And that would give you an idea of where cancer started if you didn't already know. In his case, we already knew. But that's what happens because tumors get very, very, guys, they get very vascularized. Like I said, angiogenesis, which would be the creation of blood vessels. And then those little cells cross that barrier into the blood. And once they get into the blood, they go and land in other parts of your body and they just start dividing like crazy. And that's how you can start out with breast cancer that becomes bone cancer. You know, and they only but so much surgery they can do. Like if it's breast cancer and it hasn't spread, you can take out the tumor or you can take off the breast or whatever. Or like in my uncle's case, they took out his kidney. But at that point, we didn't know if it had spread or not because we didn't see anything else. But later it was visible in bones and in brain. And those aren't things you can take off. It was your hand, you could take off your hand, but it was in his hips and his spine. You can't take that off. So you end up having to do things like chemo and radiation to try and control the cancer cell growth and keep it from spreading too fast or getting to other parts of the body that make your quality of life even worse, you know? So we like the video said, there's already R&D going on to try and target more specifically the actual processes that go on in those cancer cells to stop them from dividing instead of just doing what we do now, which is like, well, let's just throw a broad spectrum treatment at it, which does damage healthy cells along the way. Like if that's why people in chemo lose their hair, because those cells that grow, the follicles that actually grow hair get affected by the chemicals. The healthy cells get affected by the chemicals in chemotherapy. So let's talk for a bit about differentiation. All right, so this is where we start talking about multipotent, totipotent, pluripotent, all that good stuff. So when you whacked your little flatworms in half today, now there was a question on your lab about, do they have internal structures and what evidence do you have that they do? Did anybody get to that question yet? So if you looked at mine, especially like under that good stereoscope with the light, you can probably see they do have some internal structures and they have those little eye spots, which we talked about. And then they have that little mouth, esophagus, anus thing that pops out and they eat and poop with all the same thing. Awesome fun, right? But obviously those are different structures. So they're composed of different cells. So those cells have differentiated. So if we go from one cell to many cells, those cells are going to have to differentiate. They're going to have to turn into different cells along the way because we all start as one cell with the ability to become many, many types of cells, right? So animal development happens in stages. First, you have fertilization, which y'all are familiar with. You've got your female gamete, which we call an egg, and your male gamete, which is technically a spermatozoa. That's one. So gamete is the word for sex cell. When we talk about gamete, that's what we're talking about. Trees have gametes, you know, gametes are just sex cells. And then you have a blastocyst development and blastocyst implantation. And we'll look at some pictures of these and some embryo development. And then fetal development. But you don't have a fetus till you get through all these stages. You have to go through these stages first. And it's not till here that it starts to look like something you recognize. So this is pretty cool. I wanted to show you this because this is absolutely one of my favorite videos. There's a couple similar ones. This is a really good one. So when a human egg is fertilized, there is actually a chemical reaction that occurs, and under the right conditions, it can be observed as a flash of light. There's literally, the moment life begins is a literal flash of light, which is pretty amazing. Like that is the exact moment that a unique individual is created, genetically unique individual is created, is in this flash of light. So you can watch it happen because they've done a great job filming it here. So these are actually human embryos or human zygotes at this point, but they're doing it like in vitro fertilization. And they do see it in other animals too, yeah. That's the chemical reaction under different observational conditions. but that's wild. You can actually see it. So when they do in vitro fertilization, they don't always look at it that way. It's not necessary for the process of development. But do you know what in vitro is? It's when they'll take sperm and egg that are outside of a mammal. Typically, we're talking about humans, but it's a form of like reproductive therapy or reproductive health. There are some people that have trouble for whatever reason, like conceiving babies the old-fashioned way. So they'll go to the lab and they'll actually take a sperm and an egg, and they'll either put them together in a dish, and literally like you had today, a petri dish, and they'll let the sperm find the egg. And sometimes they can actually select the sperm they want and inject it into the egg, which is really kind of cool. And I've got a picture of that too at one point. So you have fertilization, and that's the process of combining your male gamete and your female gamete. So your sperm or your ovum, your spermatozoa or your ovum, and they produce your first product, which is called a zygote. So your very first thing is a zygote. So a stem cell, and your zygote is a stem cell. It's a cell with the unique ability to develop into specialized cells in different types of cells, right, in the body. And in the future, we can use stem cells, we're hoping, to, fix all kinds of problems, replace lost body parts, help with diseased processes. So these are totipotent cells, which means they can form all of the cells in the body plus your embryonic stuff. So plus your placenta and your amniotic sac, which pluripotent and multipotent cells can't do. Only totipotent cells can do that.
Yeah, there's no space in between extra and then embryonic.
No, it's a word. Extra embryonic is a word. Fun word. Extra embryonic meaning outside of the embryo. So what the embryo needs outside of it, which would be in mammals, a placenta and an amniotic sac. So embryonic cells within the first couple of days of divisions after fertilization are the only cells that are totipotent. Pluripotent cells can give rise to all of the types of cells that make up a body, and embryonic stem cells are considered pluripotent. So zygote cells are totipotent, and once you pass from the zygote to the embryo stage, there's a point where they become pluripotent. And then multipotent cells can develop into more than one type of cell, but are more limited than your pluripotent cells. And adult stem cells and cord blood stem cells are considered multipotent tissues. So we talked about, actually, I believe cord blood is actually pluripotent, as I was saying earlier. But a lot of people are banking their baby's cord blood now. So this just shows you, sorry, this right here would be your very, very early organization of cells within an embryo until it turns into something that looks like that. And pretty much most embryos, whether reptilian, whether fish, whether mammal, whether bird, start out looking a lot like that, a whole lot like that. So fate mapping. So fate mapping is a very, very cool thing. So you start with one cell, right? It's your zygote. It divides into two. Now it's an embryo. It divides into 4, it divides into 8, it divides into 16. There's a point where it becomes something called a morolla. But right before it gets there, when it's still in an embryo stage, before it gets to the morolla stage, you can go in, and this is great to do with amphibians, because with amphibians, their embryos develop outside of a body, right? You can put them in a petri dish and watch them grow. You've seen tadpoles, right? Like in the water, in a pond or whatever. So you can take certain types of dye that are bioadhesive that'll stick to living tissue. You can actually touch that dye to one of those, maybe 16 cells or 8 cells or whatever you have. Just tap the dye to it, which is hard because it gets in the water too. Tap it on there and watch. And as that cell develops, you will observe what tissues come from that cell. So whatever that cell is programmed to become is called fate mapping. It's fated to be whatever. And you can watch it. It's a neat thing I did in college. We used a dye called Nile blue, which is a nice royal blue color. So it's really dark and you can see it clearly. And we would use frogs and we'd touch that one of 8 cells, and you could see whether the one you touched happened to become neurologic tissue, digestive system, integumentary system. It'll turn into something. And it's pretty amazing to see where it goes, because those tissues will keep that color for a long time, nearly forever. Pretty neat. So here's what I was saying. You have your zygote, 1, your embryo is 2, 4, 8, and then you get to this Morula stage. Then we get to this cool blastocyst stage. This is when stuff really starts to get down to business. So if you notice, the size so far hasn't changed until you get to like right around in here. The size of this little guy has stayed roughly the same, but the cells have gotten smaller and smaller and smaller. So an ovum, a human ovum, for example, or even a mammal ovum is much bigger than your average cell. If you think about the size of a little sperm compared to the ovum, it's way huge, right? So, and the sperm are itty bitty, but you've got this same amount of cytoplasm that's been divided up, basically. Now your DNA is replicating every time, your organelles will produce new ones, they'll get divided up, all that good stuff. But right here is when it gets super cool. So most of the cells are gonna go pack themselves into one corner, and the rest are gonna form this nice, like a dome. So this is a cross section, it looks kind of like a basket, but you could think of it more like a snow globe with a bubble in the top, and the glass would be kind of like this outer ring of cells, okay? And then it will hatch. It loses this initial membrane. And then when it gets to this stage, this is in mammals when it would implant, but this is when it really starts to divide and grow. And something really cool happens not too long after this, where you can see the first big developmental change, and the cells on top. At this point, they've divided to the point where you can't really see individual cells anymore. It's like a kind of like a cohesive mass. But the first major phase in development for, I think, yeah, mammals and, well, I know mammals, but I think birds and fish, and they do it too, reptiles, is this really interesting thing where it will fold in on itself like this. It just folds in on itself. And when it folds in on itself, that groove right there typically is where your spinal cord will start to form if you've got that going on. And it will differentiate into layers. And each of those layers will become a part of some system. You can tell at that point what's going to be nervous system, what's going to be skin, what's going to be digestive. It's pretty neat. And that's one of the other first things that forms is you'll get what's called alimentary canal. So you'll just literally get a hole that goes through the middle of it like a tube. And that is your like mouth to anus canal for whatever organism is being created. That's one of the first things. So this is a human embryo under a microscope, and you get to see it go through a lot of those first stages. So this is really, really interesting.
Human embryos are dynamic. The journey of human embryos, as seen under a time-lapse microscope, begins like this.
So the clock will show up at the bottom. You can see how long it
As
takes.
shown here, is fertilized by either conventional IVF or intracytoplasmic sperm injection.
So that would be conventionally, and this would be when they actually inject.
This egg has been fertilized. At 16 hours post-fertilization, the egg highlights signs of fertilization as the male and female DNA come together. At around 24 hours post-fertilization, the fertilized zygote undergoes the first division to form a two-cell embryo. The embryo continues to divide, and 2 days following fertilization, the embryo reaches the four-cell state. And on day 3, it becomes around an 8-foot stage embryo. The embryo grows further, dividing from 8 cells to 16 cells, and from 16 cells to 32 cells. The embryo then makes the transition to reach the morula stage. The morula stage embryo progresses. Early signs of cavitation are seen. Blastocyst formation takes place.
See, there's your blastocyst where it's It's got the, almost like the bubble inside, right? Like I was saying.
The embryo is at blastocyst stage on day 5. At this stage, the process of implantation begins.
When it was in black and white at the very start, it did actually look like an egg yolk.
An egg yolk,
Egg-
yeah.
Egg whites around.
An egg whites and egg yolk. So these will obviously be, you can come back and look at your answers, but we can look at them real quick and see what we've got for your homework or your binder work. And then I do have also for you guys your test prep for this week too. So cells size increases and its amount of DNA also increases. No, its DNA is going to stay the same, right? It's just going to get big. That's why it's got to divide. And then with this one here, the smaller the cell, the larger the ratio of its surface area to its volume. I think most of these probably weren't any you had trouble with, were they? Did anybody have trouble with any of these? Y'all got through your binder work good? Okay, all of it's here and available to you. Test study topics. Make sure you have your pencil out or your camera, which however you're going to go about it. And of course, I will share this on email too, but helps to have it. All right. And she's out again. I'm telling you. Say, do you want to write down your test study topics? Yeah, maybe. Okay. So let's see. Basic cell structure. You gotta know your basic cell structure. All cells have membranes. All cells have cytoplasm. They don't all have a nucleus. All this kind of stuff, right? Organelles, what they do. Organelles and what they do. Y'all are so weird. I mean, just like
Don't look at me.
when people think of homeschoolers, like this is what they see.
I know, you're
This
giving
is
us
what
a
they
bad
picture,
name.
for real.
The person who won't stop talking about brownie points, and wants brownie.
Ah.
Smart, but they're also.
So passive versus active transport. Now, somebody remember the difference here?
When you're passive, you just sit on the bus without things. When you're active, you actively start fights on the bus.
That would be good for buses. How about transport over cellular membranes? What's an example of a passive transport? Think about the model cell we built. Osmosis and diffusion are good examples of passive transport. Active transport requires what?
Oxygen.
Huh? Energy, yeah, energy. Active transport requires energy, and it's very often through those channels, right? Through the like the protein channels, the gate. Remember all the gated channels and that kind of business, all that good stuff. Diffusion and osmosis; those are your passive examples. All right, unicellular and multicellular organisms. Remember that unicellular organisms can be prokaryotic or eukaryotic, but your multicellular organisms are going to be eukaryotic. How about organization? How do you get from a cell to an organism? Who remembers? There was an image of a dude with a fig leaf.
Oh.
Let's think.
Newton.
Start with cells, and then what do you have?
The tissue.
Then tissue. That's right. What's after tissue?
Oh.
Oh.
Organ,
Organ system.
organ systems,
Person.
and an organism. Very good. Yes, that's what we want.
Oh.
The fig leaf guy. Remember the fig leaf guy? All right.
I was thinking apple. I'm like,
ATP. What's ATP?
And protein triphosphate.
Yes. And what's it for?
Energy.
It is our source. All living things use ATP as the source of energy. What is ATP produced from? It starts as
Sugar.
ADP. And what's added to the ADP to make it ATP?
Phosphate.
A phosphate. Exactly, right? You add a phosphate, you get ATP. Do you need ATP to make ATP? Yes, you do. So some of your energy that you're using to make energy is going to get used to make energy. Yeah,
And where did the first ATP come from?
that's a great questions, aren't they? This is why I love biology. Where did the first ATP come from? I don't know. It's pretty cool though.
First.
Photosynthesis. Okay, photosynthesis takes place where?
No.
James has got it.
Well, just knowledge.
Yep. In the chloroplast, in the grana, on a thylakoid membrane, that's where you're gonna find your chlorophyll, right? And that is photosynthesis. How about respiration? What's the difference between, or what's noticeable between photosynthesis and respiration chemically? They are opposites, chemically opposite reactions, right? What goes into photosynthesis?
Carbon.
Carbon dioxide and what?
uh,
And water. And so that's what? CO2 plus with sunlight yields what?
Glucose and oxygen.
So think about it. Is a plant going to put out oxygen?
Yeah.
There you go. Plant's going to put out oxygen. Very good. So you got it. Now, what's the glucose chemical formula? C6H12O6. So CO2 plus H2O with sunlight yields C6H12O6 plus O2. In reverse, cellular respiration, aerobic cellular respiration, specifically aerobic, meaning requires oxygen, is going to be O2 plus C6H12O6 with heat is given off, and it's going to yield what? CO2 plus what's left? H2O. You got it. Awesome. Steps in cell division, what were they? Starting with an I. I punched my annoying tennis coach or
I
whatever.
can tell you the.
In-
mnemonic device.
What'd you say, Bailey? Interphase. Prophase. Cytokinesis. Very good. You're going to want to know those, what order they're in, what happens in them. Taxonomic hierarchy. Do we remember this? We start with domains now. We don't start with kingdoms anymore. We start with domains, and what are they? Taxonomic. This was how things are categorized. We start with domains. Do you remember what they were? Yeah, that's when you get into, yes, so kingdom, yep. But what came before that? It used to just be kingdom, phylum, class, order, family, genus, species. We used to just go with 5 kingdoms, but now we have 2 domains that are on top of that. Do you remember we have eukarya and archa? Remember, and the archabac, it's archaea used to be archaebacteria. Archaea is now just all the like predominantly prokaryotic stuff. Some of it's chemosynthetic. Do you remember that? We talked about like the ancient bacteria that live down in the deep sea, vents and thrive on nitrogen or whatever. All right, and then you've got after that, kingdom, phylum, class, order, family, genus, species. Do you remember the mnemonic for those? King was right. Philip came over for great spaghetti. King Philip came over for great spaghetti. And you just have to remember that archaea and eukarya are above it. They're the new ones, right? Kingdom, phylum, class, order, family, genus, species. That is Linnean, except for if you take the domains out, then it's Linnaeus. It's Linnean taxonomy. All right, what's binomial nomenclature? Who knows that one? It has to do with the taxonomy. How do we identify an organism?
Genus, name, species.
There you go, genus and species. Why do we do it that way? If I was going to be scientifically researching dandelions, why wouldn't I call them dandelions?
Different types, different names and different.
Yeah, people call them different things, different names in different languages. Shoot, we have things growing out here that people call 12 different things and they all speak English. They just call them different things. Yeah, I mean, my neighbor said something about red bugs. He's like, there's red bugs back here. I'm like, what the heck are red bugs? He's like, red bugs. I looked at him, he said, chiggers. Do you know them as chiggers? I said, I know what chiggers are. I didn't know what red bugs were. So, I mean, same things, right? So we would call it by its genus and its species. And oddly, if you've ever looked at a chigger and a clover mite, they are nearly identical. and really, really hard to tell apart. Maybe I'll find a picture of them for you. They're pretty cool. Oh, that's on there twice. Whoops. All right. Producers, consumers, decomposers, scavengers, chemotrophs, autotrophs, etc. All right. What's a producer?
Algae.
That's a good example. Yeah. Something that makes its own food, makes food for others, all that good stuff. All right. What's a consumer?
Tiger.
Now we are. Yep. Okay. Do you remember first level and second level consumers? First level consumers are going to be herbivores, right? And then you get second level. And what kind of animals are at the tippy top of that pyramid?
Apex predators.
Yeah, and what are they always?
Carnivores.
Carnivores, very good. Do you remember that if you're looking at the triangle and you have the wolf at the top, that all of the grass at the bottom is needed for that one wolf, right? Like all the grass that feeds the, whatever the wolf ate, you know, all of that, whatever's in order there, the snake, whatever it is, I don't know. Excellent. What is an example of a chemotroph? Ooh, it's a tricky one.
Worms.
Yeah, those kind of things. Yeah, organisms that live off of chemicals, right? How about a decomposer?
Deep-sea worms that live off of chemicals.
Yeah, what's a good example of a decomposer?
Mushrooms.
Mushroom, fungi. Here's the big point with decomposers. When we did our lab 3 outside, y'all with me? We did lab 3 outside. There was a question. It was like, hey, what about decomposers? If there were none, what would happen? Everybody got the first step right. Dead bodies would just pile up everywhere. That's the first part. The other part that I wrote back on pretty much everybody's lab was, and nutrients would not get returned to the ecosystem. That's the other big thing that decomposers are going to do. They're going to return nutrients to the ecosystem. Without them, we don't get nitrogen fixed, and there's a bunch of other issues that we'd have. And we said what autotroph. So what's an autotroph versus a heterotroph?
One produces its own food, the other requires to go get out and get food from others.
There you go. Excellent. All right. What are the differences between plant cells and animal cells?
Plant cells.
That's one. Yeah.
The cell walls and chloroplast.
Yep. And there's one other thing that can easily identify plant cells. Plant cell walls,
Square.
chloroplasts. There, yeah. Do you remember what it was?
Cell unit? No.
Well, that's what the cell wall is composed of. You're correct. Remember that big central vacuole? There's a big vacuole in the middle. Do you remember? And we talked about it contains water and it changes size, and that impacts whether the plant is like flopping over, standing up tall, all of that's really important. Gets into turgor pressure. Okay, we did prokaryotes and eukaryotes. We talked about that. Remind me again, what are you?
We are a.
You are eukaryotes. Very good. All right, coming up soon. It is October. October means Tinker Day. For all the new people, somebody want to tell them what Tinker Day is?
Didn't have it last year.
No, because nobody had it last year and I was heartbroken. Who can tell our new folks what is Tinker Day? Come on, people. All right, Tinker Day is a holiday celebrated at my college. It happens in October every fall, except last year because there was a pandemic and they didn't do it, and I was heartbroken. You don't know when it is. The Dean of Students decides when it is. So it's always a surprise. When it's Tinker Day, they cancel all classes and everybody dresses up funny and hikes the mountain. And before you hike, you go to the dining hall and you eat Krispy Kreme, which comes by the truckload. You hike up the mountain, you eat fried chicken and tinker cake, you sing songs, you go back to your life. It's the most fun thing ever.
It seems like it's kind of like
Best-
testing, filling you up with.
It's the best
Krispy Kreme
holiday
and then
ever. Yes.
Basically, how we would do it, like get a box of donuts and then go hike up Sally Mountain or something. like
That's what I've done almost every year.
that. Okay.
Yep. Uh-huh.
I've never done it. so-
Oh, yeah. Well, I usually, we have donuts, and then if we can swing it, if schedule allows, the boys and I have gone up Sony Mountain, and I will go pick up fried chicken from Publix and pick up some piece of cake that looks close to Tinker Cake, and we go, we do
Are
our
you gonna
thing.
take all
So,
three of us up
Should
at the
Tinker Day fall on a Tuesday, we still have to have class, because I can't be like, oh, no class. But regardless of when Tinker Day falls, either on a Tuesday or after a Tuesday, I will have donuts for you that day or the following day. There will be Krispy Kreme on Tinker Day. I will send an announcement. Oh my gosh, it's Tinker Day. And if you send me a picture of yourself wearing green and gold, you will get extra credit on your next test, because that's how I roll. Yes, because those are our colors and it matters. And Tinker Day is the best holiday on the planet, hands down. Most fun ever.
She's brainwashed.
I mean, if
It's
I-
the best.
If you get off from college, that sounds
Yeah,
cool.
you don't have to shop for anybody. You shop for yourself. You go and you raid Goodwill weeks ahead of time. You spend September getting, you do, because you want the cost, you want that craziest outfit. The best was my freshman year, my friend Amy, she got a shirt at Goodwill and it was like a mechanic shirt from a garage, like the blue kind with the embroidered patch, and it said Mitch. And we were calling her Mitch all day. And like we hiked all the way up the mountain and we were on the way down. And for some reason, she stuck her hand in the breast pocket of the shirt and pulled out a toothpick. And then we're all like, was it Mitch's? Did he use it? I mean, who knew? But it had been in there forever. And it was awesome. But everybody dresses up funny and people plan their outfits like months in advance. So all the green and gold, and we're good to go.
What are you doing?
I have so much.
I have a single straight that
I
is
have
green.
one green shirt in my
I
entire
just got
room.
rid of so many green shirts.
It is the best.
I
And
just-
last year they didn't do it and it was just heartbreaking. There was only one other time that they didn't do it. And this is since 1840. When did Tinker Day start? 1860 something. I have
There
the
was
date somewhere.
Krispy Kreme in 1860.
Well, they didn't get Krispy Kreme then, but they,
Hey,
I'll tell you
don't
when it
have to
started
spoil
a long time ago.
What?
It's been a long time thing.
it.
It's a long time. Tinker Day is a long, it's a very old holiday. These are your teams for next, but I have to change it because the Sabins are gone. And I think we're going to be getting our new cool dude, Anthony, who is very cool. I really liked him. Super nice kid.
I'm surprised he wants to come back. I really am.
You guys are funny.
You gave your kid the most, like,
I knew he'd fit in over there.
And we let him cut a small.
gave him the
thing.
most intense team in the entire.
I did, but
Hey,
it's
no, no,
called
you know what
baptism
she
by
did?
fire.
She gave.
All right,
she gave
here,
him
let's
the
see.
fun team.
Here's a picture of Tinker Day.
Fun for us, at least.
So there were two of them in the 1880s, and it became an official holiday in 1895. And this is the famous, one of the famous old Tinker Day pictures, and this is why it's freaking awesome. So this picture is from 1898, and those ladies are no joke, climbing a mountain in wool skirts with leather shoes that probably have wood in the heels, like leather boots. Check that out. These girls are no joke. There used to be a giant picture of this in the science building, so I saw it every day. So yeah, there's your lab teams. Start thinking about whether or not you're going to volunteer to be captain. And here's your, in your
Like, he
last
wants to fail.
5-10 minutes, I want you to take out your binders in the last 5-10 minutes, take out your binders and make sure that you are organized. Take out stuff you don't need. Put everything back where it goes.
She immediately said yes.
You are, hold on,
Yeah. I thought.
you're there again.
I think, okay.
Test them again. So I got to fix that, obviously. Oh, progress reports. I forgot to mention that to the other group. I'll send them out probably tomorrow, if not today. So take the time to make sure your binder is put together properly, that your Q2 stuff is installed, and most importantly, that you remove your old lessons 9 through 17 and replace them with new ones, because your old lessons 9 through 17 just said like whether or not there would be homework. It didn't say what the homework was. Your new lessons 9 through 17 do say what the homework is. So you're going to want to fix that. Take those out, put new ones in. And I think there's nothing past this, right? No. Okay, hold on one second. All right, I'm going to tell Michael goodbye then. Goodbye, Michael.
Oh, bye-bye.
Feel better.
Bye, Michael.
See you later. Bye.
Bye.
Y'all can